we will go for primer designing. So when we need to do PCR, okay, primer designing is the important problem while doing a PCR, because we usually get primers maybe from publications. Sometimes that publication primer may not be working for us. Mostly we need to design our primer, and we need to synthesize it by giving to synthesizers, DNA synthesizers, and they will synthesize and give. Or maybe you people, if you have a DNA synthesizer in your lab, you can synthesize yourself. So primer designing is an important tool while you are studying a particular gene. So you should follow some rules while designing a primer, that we will discuss first, and then we will design a primer using Primer 3 software. That is what we are going to study next. Designing a primer for PCR. A primer length determines the specificity and significantly affects its annealing to the template. So the specificity of the primer towards the template is determined by the primer length. Okay. So primer length should be optimal. Primer length should not be so high or should not be so short. Too short means low specificity will be there and non-specific amplification can occur. Too long means decrease in template binding efficiency at normal annealing temperature due to high probability of forming secondary structure. If it is so long, they can form hairpin like structure. They can themselves form hairpin like structure, so that they will not bind properly to the template. If it is too short, they will not specifically properly bind to the template. So this is the major thing you should think. So the primer should not be too short or should not be too long. This you need to keep in mind. So there should be an optimal primer length. The optimal primer length should be 18 to 24 base pair. It should be 18 to 24 base pair. And in PCR, the optimum amplicon size also should be around 300 to 1000 base pair. Avoid greater than 3 kb. So the amplicon, the product we need to get after the PCR also should be in this range, 300 to 1000 base pair. Avoid more than 3 kilo base pair. So this is the first important thing. The primer length should be 18 to 24 base pair, and amplicon length, the optimal amplicon length should be 300 to 1000 base pair. First thing you should understand while designing a primer. Second rule, the melting temperature. Melting temperature of the primer. The Tm is the temperature at which 50% of the DNA duplex dissociate to become single stranded. So Tm is a temperature at which 50%, if we have the 3, 4, 5, 6, okay, this much, one more, okay. So Tm is a temperature at which 50% of the DNA duplex is 50% of the DNA duplex is dissociate or denature or dissociate and become single stranded, and the rest of the 50% remains like that. So the temperature at which 50% of the DNA duplex dissociate and becomes single stranded, that temperature is called the Tm or melting temperature. This Tm of the primer will be determined by the primer length, its base composition, and its concentration of the primer. So these three parameters determine the melting temperature. Also affected by the salt concentration in the PCR reaction mix. So these four parameters determine the melting temperature of the primer. Melting temperature is determined by the primer length, what all base present, A, T, G, C, what all base present, and the concentration of the primer, and salt concentration of the primer also determine the of the PCR mix also determine the melting temperature. Usually melting temperature is calculated by this equation 280. If consider a sequence, a primer sequence A, T, G, G, C, T, G, G, G. This is how many sequences are there? 3, 9. Okay, I will add one more G here. Okay, so how we can calculate the suitable only for 80 or shorter. So this equation we will do and calculate the melting temperature of this. Okay, 2 into number of A plus T, number of A plus T, 1, 2, 3, plus 4 into number of G plus C, 1, 2, 3, 4, 5, 6, 7. So it will be 6 plus 28 as 28. It will be the melting temperature here will be 34 degree will be the melting temperature for this. It's about 10 nucleotide only. When we calculate for 18 nucleotide, the optimum nucleotide number is 18 to 24. So you will get the correct melting temperature. So this is how we will calculate the melting temperature of the particular primer. Okay, hope you understood. So optimal melting temperature, usually melting temperature will be between 52 to 60 degree. TM above 65 degrees generally avoided because of the potential for secondary annealing. Secondary annealing chances are there. So we will not use this temperature of 65 degree. Higher TM, the higher TM of 75 to 80 degrees recommended for amplifying high GC content targets. For high GC content targets, we use higher TM because GC bonding is very high to get very high temperature is needed to get denatured compared to that of the AT. So this is about the melting temperature. How to calculate the melting temperature, what melting temperature is the optimum, and mostly the optimum temperature is 52 to 60 degree. So next, the rule of primer designing, the specificity and cross homology. Specificity and cross homology. This is the next primer designing rule. Specificity means determined primarily by the primer length as well as the sequence. So the specificity of the primer is determined by the primer length as well as sequence. The adequacy of a primer specificity is depend on the nature of the template used in the PCR reaction. So adequacy of the primer specificity is depend on the nature of the template used in the PCR reaction. So it will depends on the specificity of the primer depends on the template used in the PCR reaction. The second parameter is the cross homology. May become problem when the PCR template is genomic DNA consists of mixed gene fragments. The primers contain high repetitive sequence which are prone to generate non-specific amplicon when amplifying genomic DNA. When amplifying genomic DNA, primer can bind to primers containing high repetitive DNA can bind to different regions of the genome and produce non-specific amplification amplicons. So this is also you should keep in mind, while cross homology should also be prevented. So two rules are specificity, the primer length as well as the primer sequence determine the specificity, and this primer specificity is depend on the sequence of the template also. Second is the cross homology, which become a problem when the PCR template is genomic DNA. When the PCR template is genomic DNA, when we are having a very big genomic DNA, 3 into 10 raised to 9 bases of genomic DNA, okay, then the primer, if the primer contain highly repetitive sequence, G, 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 C, 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 repetitive sequence, the primer can bind to any of the region of the genomic DNA and create amplicons, non-specific amplicons. This is one of the major rules. So when you design the primer, if you are using genomic DNA, you should design the primer with less repetitive DNA sequence in your primer. So these things you should keep in your mind when you are designing the primer. Next rules for designing the primer is the GC content. GC content is very important while designing a primer. So primer GC content is a very important parameters when you are designing a primer. Optimal GC content should be 45 to 55%. About half of that, if you have 18 nucleotide in your primer, about 10 nucleotide should be GC. So optimal GC content should be 45 to 50%. The common GC range from 40 to 60% GC range we need to. GC content is very important while you are designing the primer. You should keep in your mind. Repeats. Single base touches. So long runs, repeats and runs. So we will see runs first. Single base touches. Long runs increasing mispriming or non-specific aliening potential. Long runs of a nucleated, single nucleated runs stretches means if the primer contain like that single nucleated base structures, long single nucleated base structures, it increase the non-specific annealing and then G, then GC, like that. So if the single nucleotide base structures are present more in your primer, the maximum acceptable run will be a 4 base pair. 4 base pair single bases can come. After that, if it come, it can lead to a non-specific annealing. And then we will come to repeats. Dinucleated repeats, if it the AG like that, if dinucleated repeats come in your primer, repeats increases mispriming, then also mispriming can occur or non-specific annealing can occur. Maximum acceptable repeats should be 4 repeats. 4 dinucleated repeats is okay, allowed. Otherwise more than that should not be allowed. So these three things I think you understood. The GC content should be around 50%. Runs means single nucleated runs should be only 4 and repeats should only 4 repeats should only be there. So this you should keep in mind while primer designing. So general rules of primer designing. So next is the primer secondary structure. This as we told, if we go for a bigger a long primer, it can form hairpin like structure. Hairpin like a structure. It can base pair itself. It can base pair itself and form hairpin like a structure. So intramolecular interaction can occur and it can form hairpin like structure. So you should keep in mind that during primer designing, the intramolecular base pairing type of nucleotide should not be there. They negatively affect the primer template binding and lead to poor or no amplification. If the secondary structure form, it will not bind to the template and no amplification can be seen. Self-dimer, homodimer can occur. There will be usually in PCR 2 primers, forward primer and reverse primer. Okay. This forward primer itself can self annihilate. They can form dimer. Okay. So it will lead to it will dimer each other. If the sequence are in such a way that it can be self dimerized, it will not bind to the template. So we can't get the product. Or cross dimer, the forward and reverse can dimerize. So then also it will not bind to the template. So your sequence should be such a way that it should not form cross primer, dimer or self dimer as well as hairpins. And then I think the last no, one or two rules are there. So hope you are following. Hope it is everyone is going. Hope it is beneficial. Okay, everyone is going. If it is not beneficial, I am teaching. So hope it is beneficial for somebody. So anyway, I will be going through the primer designing. I need to complete the primer designing today, and we will be continuing with the NEB cutter tomorrow, and then rest of the things. Primer designing, lot of process remaining, so I need to finish this. So otherwise we will do the primer designing also tomorrow. So let us see the rules of the primer. This you need to keep in your mind. If you are not designing the primer with the primer 3, if you are designing your own, you can manually design the primer. Okay, if you are designing manually the primer also, you should keep in mind all these rules. You can design the manually also primer. So if you are designing it manually also, you should know these rules. I can't go directly to primer 3 and show you how it is. So you should know what is 6' translation. You should know what is how we design primer. So GC clamp refers to the presence of GC within the last bases at the 3' end of the primer. When we see the primer, 80 is a primer. So it starts from the 5' end, 80. So G, A. So at last 4 nucleotides should be GC. So as it is G, it can access a clamp. This is a 3' end. It can access a clamp which help in very strong binding to the template. Okay. These 3' end last 4 bases should be a GC clamp which can help in essential in preventing mispriming and enhancing the specific primer template binding. So these GC clamp can bind firmly to the, because GC bonding is a triple bonding, bind firmly to the template, thereby allowing the further base pairing and help in the binding of primer, annealing of primer properly to the template. So GC clamp is very important. So maximum 3' end stability. Another thing is the maximum 3' end stability. While 3' end stability improves the priming efficiency, too higher stability could negatively affect the specificity because 3' terminal partial hybridization included in non-specific extension. So 3' end should be of the primer should be highly stable, so that only the proper extension can occur. And the last rule is the annealing temperature. You know that when the primer is designed and that in that bottle, usually you will get it from the company. I only get from company. I have not synthesized any primer in my lab. So from the company, the TM will be marked. From the TM only, we will find out the annealing temperature. Usually the TM annealing temperature TA will be 5 degree lower than the TM. We will take a 5 degree lower than the TM. So TA or annealing temperature is determined by the TM of the both primers. So TA will be, annealing temperature will be 5 degree less than the TM. Higher the TA enhances the specific amplification, but may lower the EDC. So we should have a optimal TA. We can see the optimal TM will be about 52 to 60 degrees. So less than 5 degree of this can be the optimal TA. So it should be useful. So let us design the primers now. So let us design the primers. Hope you understand while you are designing the primers your own, or while you are doing it using any tool, you should understand this. So for that we need the gene sequence which we need to amplify. Okay, first we will design the primer, then we will go for PCR, in silico PCR with that primers. Okay, we will design, for designing the primer we need the sequence. I will tell. I will tell that for designing we have a DNA sequence, and in which this is the gene. So we need to clone the gene. So if you are not cloning the gene, if you want to for 16S etc., if you want to see some regions of that 16S RNA like that, we can design the primer somewhere inside the gene, so that we only need a part of the gene, so that we can identify the particular genome or particular gene. Just for identification we can just give the gene sequence or gene nucleotide sequence from the gen bank, and we can just take the nucleotide sequence from the gen bank, and you can paste it in the the primer the primer 3, so that you will get the your primer designed in anywhere inside the gene. So actually when we are what we need to do is that by properly to design the primer should be designed in the flanking region, nearby region of the gene, so that we will get the full gene get amplified by the PCR. So the primer should be outside the gene, nearby the gene, the flanking region of the gene, so that we can get the full gene amplified. So that we need to get some sequence upstream and some sequence downstream of the gene which we are going to target. So some sequence upstream and some sequence downstream. For this we use UCSC genome browser. From this browser we can get the sequence with flanking region which we specify, some sequence upstream and some sequence downstream. So we are going to first take the sequence from the UCSC genome browser. Just follow me. Okay, where is that? We are going for UCSC genome browser. So you go to UCSC genome browser. You can go here. Click the genome browser. Here you can see the tools, you can see genome browser. In the tools you can see genome browser, and we need to go to the Asian genome browser page. Here, take me to Asia genome browser. So you will get a page here. You can see the latest assembly of human genome assembly. I think one more assembly, it is not. You can say that see this 2013 assembly. What assembly it is? So I need to check T2T. You can see take this is the default assembly we are showing. So you can see take the 230, because this genome sequence has been sequenced from 2003. Every year the sequence has been, the assembly has been updated. So we will take the latest sequence assembly. From the genome sequence we can find out the gene, we can give the chromosomal region etc. we can give to find out. We will take a gene only for our PCR. We will take a gene called GAPDH of GAPDH. We will take GAPDH 17 here. We can take, you can take your own sequence. So your own sequence, you can search any gene sequence. You can see the properties of the gene, all these GapDH gene. You can see the GapDH gene here. You can GapDH17 gene. You can see the properties, everything. We are not going for a UCSC genome browser in this workshop. So you can download this sequence. Okay. While downloading, we should have a we are just downloading the GapDH sequences. We can take GapDH sequence from the nucleotide database, GenBank from the NCB also. But here, what you can do is that go to view and click DNA to download the sequence. Okay. Then here, what we can do is that we can add one 500 base pair upstream and 500 base pair downstream of our gene, so that we can design our primer in this 500 base pair. So the primer will not touch our gene. So we can download a sequence with a 500 base pair upstream and 500 base pair downstream. So before that, you should look on the number of base pair present in the gene. It's 1001. Okay, this is important. I will show you what to do. 1001 base pair is your gene size. Okay, we will go to DNA and we are going to download the sequence. For that, we need to take a 500 base pair upstream and 500 base pair downstream, so that the gene will be coming outside our the primer will be coming outside our gene, so that our gene will not be affected when designing the primer, so that we will get the full gene isolated. Get DNA, click the get DNA. So we got the sequence having 500 our gene will be coming in middle 500 base pair here upstream, 500 base pair here downstream, we have just taken, and our gene will come in middle. Okay. So if we get the primer, one primer, the forward primer here and reverse primer here, we will get the whole gene get amplified by the that using that primer. So we are just designing the primer now. Just copy this sequence, and then we will go to the tool primer 3, which is used in designing the primer. Primer 3 is the tool which is used in designing the primer. So in this primer 3, you can just copy our sequence. So our sequence is here in the primer 3. So then now we need to pick the forward primer as well as reverse primer, that is seen default. And next thing we need to give is that our sequence is about 1000 base pair only. Mostly the primer 3 can pick the primer anywhere in the forward primer, they can pick here, and reverse primer they can pick here. So we will not get our gene amplified properly. We will get some piece of DNA which belongs to that upstream overhang. So either sometimes the primer 3 can design forward primer here forward primer here and reverse primer here, so that we will get a sequence here which doesn't code for the our gene. So in order to prevent that, we should protect our gene. First we should exclude our gene. Okay, no primer should be. That is why we have not taken the sequence from the NCBI. So first you need to exclude our gene so that no primer should be designed inside our gene. So only we have gone to UCSC genome browser and taken 500 base pair upstream and 500 base pair downstream. So now we need to exclude our gene. How to exclude the gene? We should add from where our gene start, we have added 500 base pair upstream. Okay, 500 base pair upstream. So our gene start from the 501 sequence. So just add 501 and comma. What is the size of our gene? Size of the total size of our gene is 1001. It will come because I have already done it. So 1001. So total size of the gene plus where our gene start. That region need to be excluded from the primer designing. Okay. Then also we have a problem that I first told. Primer can be designed in the forward and reverse primer can be designed. We have excluded the gene, okay, but the primer will not be designed in the gene. But actually the problem is that the primer can be designed in the left overhang or right overhang. It will not be designed in the gene, but the forward and reverse will be designed in the same overhang, left or right overhang. So it will not get our product. So another tool is there, coming down. This we can keep optimum because they have given the primer size optimum 18 to 20, 18 to 23, all optimum, or standard the primer parameters they have given. We can keep it as. But here is a product size range. What is our product our gene size? We can give that range here, so that if this is our 500 base pair only, this overhang, okay. So if they design forward and reverse primer here, the size of the product will be less than 500. So our it is less than 500. If it is designed in the in this overhang, it will also be less than 500. So our gene will not be there. So our gene size is 1000 base pair. So we can give the size of the product around 1000 to 1200. One range we can give. So if we design a primer like that, our gene should be seen in between the forward and reverse primer. After doing this, we can click pick primers here. All other things we can keep it as default. Pick primers here. Yes, 2 primers they have picked. One you can see, this region is our gene region. They have excluded in designing the primer XXXX. So in our gene, inside our gene, they have not designed any primer. Otherwise, if you have not given that parameter, they would have designed the primer here. Okay, they have excluded our gene, and they have designed the forward primer here in our overhang. 500 base pair we have taken overhang. In the overhang, they have designed the forward primer, and they have designed the reverse primer in our left overhang, right overhang. So you can see the forward primer and reverse primer is synthesized. Okay, you got it? Otherwise, you can just check the recordings. So hope you got how the primer is designed. So for designing primer, you should because when you are cloning etc., when you are just for gene identification, you can just take it and paste it in primer blast or something in NCBI, and you will get the primer, and you can design the primer. If it is inside the gene also, no problem. When you are going for cloning etc., we need to get the full gene. Okay, for full gene you should design the primer on the upstream and downstream, the flanking region, upstream and downstream of our gene. So you first go to UCC genome browser and download the gene with 500 base pair upstream and 500 base pair downstream, and then you give the parameters like that. Your primer should come 500 base pair in the forward primer should come in the upstream sequence, and the reverse primer should come in the downstream sequence. You can just look at the recordings, so how it is done. So you can give that parameters so that the primer should be properly designed in the upstream and downstream the overhang. So you will get the correct primer in the correct region. So now now we are not going for giving that primer to the company to design. Before that we can just go for a PCR also. Do a PCR. For doing a PCR, the UCC genome browser also has a is in silico PCR. You can directly go from here, or you can go from UCC genome browser. I will end up with that. You can go for a in silico PCR. We have designed a primer, and we have the genomic DNA isolated by phenolchloroform method in our append of, and we are going to use this primer to find out which gene we have taken, GAPDH. So GAPDH, whether our primer specifically, we have designed with all these parameters which we have studied, specifically bind to the GAPDH and amplify the GAPDH. We can see, okay, the in silico PCR. In silico PCR, we are going to search against the human genome. Okay, GAPDH human only we have taken, so we are going to search against human genome. We can take other genomes also. If we have done the work in other mouse or any other, you can take mouse is there, African frog is there, African cloud frog, alpaca, I don't know, very baboon is there, kiwi is there, and many of the organisms are there. Caenorhabditis elegans is there. So like that, many of the organisms are there. So we can, as we have selected humans, this is the latest human genome assembly, and we can give our forward and reverse primer here and check whether it will amplify GAPDH or it will bind to some other part of the genome, and which will go for any mispriming and give any unwanted product. We can just take our forward and just take our reverse and just paste it here. I have already done, I usually do this works before I come to the class. So actually I have already done all this, and then only I can teach. Okay, so just submit this. So now you will get the amplified region. This region is got amplified by in silico PCR, and whether by using this forward and reverse primer, and they have used this temperature also 61.8 and so 59.8 as the annealing it's annealing temperature. Annealing temperature calculation done by salt concentration this much, and 5 nanomolar oligo concentration, and code calculated the melting temperature comes from this code. The code to calculate the melting temperature comes from. So we use this much of annealing melting temperature. Sorry, this is melting temperature. This much of the melting temperature of the primer. So you can use the annealing temperature according to that, and they have used and get this amplified product. Then we will have a confusion whether this is GAPDH or not. Here there is a link to the same UCSC genome browser. Where is the genome browser? This is the genome browser. Is it from where we taken? You can go back and see where is this gene present, this GAPDH. It is present in the chromosome Y 2861. That is the region where it is found. You can see from here itself 2861 to, so you can click here and see GAPDH. So you click here and then you can see it is GAPDH17 only. So the gene which we have amplified is none other than, we have designed our primer, and gene which we have amplified is none other than GAPDH only. So this is how. So we confirm it using in silico PCR also, and so that we can find out that our primer design is correct. So you give it to the people who synthesize it, so that you can do it in the in vitro condition. Hope you understood these two tools.